Hello everyone, myself Dr. Suresh, Professor of Biochemistry. In this video, we'll talk about the fatty acid synthase complex. Before studying in detail about the proper fatty acid synthesis, it is mandatory to know the details about the enzyme which is involved here. So, it is a single enzyme which is involved in fatty acid synthesis, that is fatty acid synthase, and it is calling as a fatty acid synthase complex. So, sure, let us go through the few facts regarding the fatty acid synthase complex. The system, you can make out that this is a multi-enzyme complex. So, the other examples what we have discussed in our previous videos regarding the multi-enzyme complex, such as pyruvate dehydrogenase multi-enzyme complex, popularly PDH, and alpha-ketoglutarate dehydrogenase multi-enzyme complex. So, there we can see 5 coenzymes have been involved, but the difference between them and the fatty acid synthase complex is here the involvement of enzymes is 7. Okay, so PDH as well as alpha ketoglutarate dehydrogenases, they have 5 coenzymes, but here the 7 enzymes which are involving in fatty acid synthase complex. So to talk about this enzyme complex, it is a dimer, and the dimer means 2 units will be there, 2 subunits. Okay, and these two are both subunits are identical, they are same. Okay, and each subunit of the complex is organized into 3 domains, right, and with 7 enzymes. So what are the advantages of multi-enzyme complex? So intermediates of the reaction can easily interact with the active site of the enzyme, so no need to go from here and to other place. So once substrate has been come to the enzyme complex, it will react with step-by-step enzymes, and then finally the product will be released. Once it is entered into the multi-enzyme complex, it will not leave until the product has been formed. Okay, and the main advantage is no need of separate genes to code different enzymes. As it is a multi-enzyme, all the enzymes are at one place, single gene codes for all the enzymes, and all the enzymes are in equimolar concentrations. So the efficiency of the process is enhanced. So time taking and all will not be there. Okay, so time consumption for the reaction or the reaction time is very less. So to talk about the first domain, as we said, it is a multi-enzyme complex, is of a dimer. So 2 monomeric units are there. So we'll talk about one by one. So first domain are a condensing unit. So it is the initial substrate binding site, and the enzymes involved are beta-keto-acyl synthase or condensing enzyme. Okay, CE is a short term code, condensing enzyme, and acyl transferase AT, and malonyl transacylase. Okay, malonyl transacylase. So first domain is divided of 3 subunits. Okay, so one is beta-keto-acyl synthase or condensing enzyme, and second enzyme is acetyl transferase, and third is malonyl transacylase. Second domain or reduction unit. Okay, so first domain is of condensation, second domain is of reduction. Okay, and we'll see what are the things has been arranged here. As we said, 7 parts are there, 7 enzymes involved in this multi-enzyme. So first 3 involved in condensation unit, and next 4 will be involved in reduction unit. So what are all the enzymes involved in reduction unit? One is dehydratases and enol reductase, beta-keto-acyl reductase, and acyl carrier protein. So remember of this acyl carrier protein. So this is the one playing major role in fatty acid synthase multi-enzyme complex. So what are there? 4 enzymes in second unit, that is reduction unit: dehydratase, enol reductase, beta-keto-acyl reductase, acyl carrier protein. Okay, so the acyl carrier protein has phosphopantothione group, to which acyl groups are attached in thioester linkage. So phosphopantothione group, where sulfur is also, actually to say, in these acyl groups, acyl carrier protein, sulfohydryl groups will be there, okay, which play major role in thioester linkage. Acyl carrier protein acts like CoA carrying fatty acyl groups. Okay, acyl carrier protein which is acting like a coenzyme A which carries fatty acyl groups. So if you go through the picture here, so 2 subunits, okay, reduction unit and then condensing unit. So the linking between the condensing and reduction unit you can make out here. So in green color there is a condensing unit, and in pink color you can make out reduction unit. Okay, so releasing unit is also there, okay, on both the ends. So here in fatty acid synthase complex, so upper and lower units are 2 monomers of the complex. Dotted line represents functional divisions: condensing enzyme, enzyme reduction complex, and acetyl transacylase, ketoacyl reductase, malonyl transacylase, acyl carrier protein, and dehydratase along with the thioesterase. So you see here in short codes we can make out here acyl carrier protein, ketoacyl reductase, enzyme reductase, AT dehydratase. So all these in short codes we have written here, and both the units are linked by you can sulfohydryl groups you can make out. So cysteine is the amino acid which is mainly helpful in linking of both the domains. So that's all about fatty acid synthase complex. Thanks for watching. Thank you.